So I'm gonna discuss two topics now within the next 40 minutes, but I'll try to make it a little bit shorter because we're running over time. First of all, manufacturing changes versus biosimilarity, a topic that has been already addressed through a question before the coffee break. And then I will discuss a bit more extensively about the issues around interchangeability and so on. And because I believe that for the real life situation, interchangeability issues are much more important than the theoretical considerations between manufacturing changes versus biosimilarity. I will keep this presentation a little bit shorter than anticipated, so I will skip a few slides. As was addressed in one of the questions, can we consider a manufacturing change as leading to the situation that an originator would become a biosimilar of its own? If we look at the different processes, I think we can decide that this is not the case, but I will come to that. First of all, let me briefly introduce what's the concept of manufacturing changes. This means that first of all, if you want to introduce a change in an existing approved manufacturing process, one should realize that the introduction of these changes is also very tightly regulated, and that you will have to demonstrate that if you introduce a manufacturing change, that this will not have an impact on the clinical safety or the efficacy profile of the product. And also, when introducing particular changes, depending on the type of change, you will have to do some kind of a comparability exercise before the change and after the change. And there are some guidelines already quite some number of years in place that everybody that wants to introduce a manufacturing change, be it an originator or a biosimilar, one has to adhere to and demonstrating through analytical comparisons that your change will most likely have no impact on the clinical profile. But obviously, depending on the type of process change, the risk that this change may introduce some impact on the clinical efficacy profile is also different. And here you see a few examples. For instance, changing a relatively small step, changing a filter supplier within your process, if you have sufficient information, of course, on the typical characteristics of the filter, I mean, this most likely will have no impact on the clinical properties, and this can also be, let's say, demonstrated in part by the analytical data. Obviously, when you change something to the cell culture process that is going on, this already may induce some more uncertainties with respect to the possible impact on the clinical efficacy. And definitely when you have to develop, in very rare cases, when a company would decide to develop a new cell line, then obviously you will need much more than only analytical data, but you may also need actually also in some cases really some clinical data to demonstrate that apart from the analytical comparability, you also have a similar clinical property. And this again relates to the type of change on the one hand, and actually the requirements that will be needed to have this manufacturing change accepted by the regulatory authorities, proving that indeed it does not have an impact on the overall properties of the product. And I will skip this one. Obviously, any company introduces changes on a regular basis to improve their production process, to improve the efficiency, and so on. So the European regulatory authorities and the other regulatory authorities are very well acquainted with the concept of a manufacturing process. For the sake of time, we will skip this one. So it's clear that also a manufacturing change has to go through the regulatory authorities and is very strict controlled, and that after a manufacturing change you can say that the product actually remains within the range of specifications, the preset range of specifications before and after the change. Now, this is very much in contrast to a biosimilarity exercise, where the purpose, the nature, the reason why you do this is completely different. Because the biosimilarity assessment is actually done during the entire process, starting from the very beginning of your process until the very end. You should also realize that, and that's a very important difference in the concept of manufacturing change versus biosimilarity assessment, is the fact that within the process of a manufacturing change, you know as a company exactly all the different steps. If you have 200 different steps, you know the characteristics of your product after each of these steps. And so it's much more easy or much more less risky to predict the possibility of a significant change or no significant change when introducing a manufacturing changes. Whereas for the biosimilarity assessment, you should realize that you do not know anything about the product except the analytical data that you can perform on the drug product that is available to you. You have no idea on the manufacturing process. You have so these 200 different steps that is known by the originator company, you don't know anything about this. You have to develop it all by yourself. So there is a huge, what we call a knowledge gap in the situation of biosimilarity assessment. And then obviously the type of exercises of analytical data of assessment is also completely different. For a manufacturing change, it's much less extensive, obviously, because you know much more to which defined parameters you have to look. For a biosimilarity exercise, you have to go through everything of the entire possible scala of experiments that you have to compare with. And this is just to illustrate visually, depending on the type of change. And here, for instance, we have a relatively, what we then call a moderate risk manufacturing change, which is supposed to have only a low risk on the change of the properties. When you have an extensive, a high risk manufacturing change, here in this case, in the case above, you do not need any clinical study if it's a low or moderate risk. In the case where there is a high risk manufacturing change, such as a new cell line that you develop, then obviously you may need some clinical studies to demonstrate this similarity. In case of a biosimilar development, you have no idea on the product history, you have no idea on the process evaluation. So this is a huge knowledge gap compared to the situation of a manufacturing change. And this just to visualize or summarize a little bit, you have to develop a new cell line. You actually have to develop your process starting from scratch in the case of a biosimilar. Within the manufacturing change, you should also realize that a company has a huge set of data already available on the product before and after the change, which is also very much important. In the situation of a biosimilar development, you do not know anything about the different steps, so you have all this information that is lacking, basically. And then what is also very important and implicitly also included in here, you have reference material after step 1, step 2, step 3, up to step 200 or whatever. In the case of a biosimilar development, your only reference point is the purified reference product, the drug product from which you have extracted your active pharmaceutical ingredient, and that this is your only reference point. You don't have any intermediate reference point to refer to. So, in conclusion, very briefly, I apologize for being so quick, but I think for the sake of time, I mean, these are definitely two distinct approaches. Whatever you may read in some papers, it's definitely, you can definitely not say that, and also if you look at the registration files, you can definitely not claim that after a number of manufacturing changes, the originator or an approved biosimilar would become a biosimilar of its own as such. So if there would be any question, I propose that we do it after or if there is a very urgent burning question, we can treat it right now. In the other case, I suggest we switch to the switching issue. Okay. Thank you. So, the obvious question when biosimilars are being introduced onto the market is, what will we do with patients that are already under stable treatment with a particular product? I think to first set this point, I think for a new patient, there shouldn't be any discussion. When your product is approved, be it an originator or be it a biosimilar, when the biosimilar is approved, you should basically not hesitate to use it in what we call a naive patient, a patient that has not been treated yet with this particular product. The question arises, how much information do we have on the safety issues and of long-term efficacy issues when we switch a patient that is under stable treatment for many years with a particular product, and then you would decide to switch the patient to the biosimilar? When we discuss this, in many papers you will read about switching from the originator to biosimilar. But you should also realize that in real life this is not the end. I mean, you should also consider then switching from the biosimilar to the originator, because there is price competition, and in certain cases we see that the originator becomes cheaper than the biosimilar. So there will be reasons to switch from originator to biosimilar, but also from biosimilar to originator. And as we will come at the end, we will have also different biosimilars. If we discuss switching, we should also consider how to deal with switching between different independently developed biosimilars, and how will we deal with that. So let me first put the context again. The question about switching is much less of an issue with generics, with chemical copies, because these are perfectly identical to each other, at least in most of the cases, as you will know. But here, just to remind you, we are dealing with products that are very much similar, but not fully identical. So there are some differences that may have no impact on the safety profile of the clinical efficacy profile if you stick to one product, but there is, at least from an immunological point of view, a possible uncertainty with respect to what happens if you trigger the patient by switching back and forward, and I won't go into detail on that. It's also important in discussions to look at the definitions, and I would like to stress that interchangeability is actually not a matter of an opinion, but it's a matter of a property of two drugs. A property, if we say that two drugs are interchangeable, this means that if we give either the one or the other product, it is supposed to give the same, to result in the same clinical effect in any patient at any given clinical setting. And interchangeability also implies that when you switch back and forward between the two products, this will also have no impact on the safety nor on the efficacy profile of the two products in any given patient. If you talk about switching, very briefly, this is actually a decision that the physician may make. He or she has a patient under stable treatment, and the doctor, the physician, may decide for some reason to switch the patient to another product, being a totally new product basically, or switching to a biosimilar. And switching is strictly spoken only allowed when we have shown that the product is interchangeable, when the two products are interchangeable. That should be, strictly spoken, the most optimal approach. And then substitution, obviously, as you may know, this is the decision of the pharmacist, and is also usually within some regulatory guidelines. This is a decision of the pharmacist to actually deliver another product than the one that has been prescribed, basically. Now, it's very important, and as Dr. Till already has referred to, I mean, within the European Union, you will see that there are the European Medicine Agency decides about the approval of the product. But as you can see, in their guidelines or in their regulatory procedure, they don't say anything about interchangeability. They decide, or they say, that the decisions on interchangeability or substitution is actually a matter of the national competent authorities. And you know, that's why I stress that interchangeability is a property of the product. So it's a bit strange that, you know, some national competent authorities would decide that two products are interchangeable, and that another national competent authority would decide that these properties would not be valid for the same product. This is a matter of taking responsibility, or in the case of the European Medicines Agency, I personally believe not daring to take their responsibility. And for questions, that's what I literally is on the site of the European Medicines Agency. For questions related to switching from one product to another, patients should speak to their doctor and to their pharmacist. So the European Medicines Agency does not want to take any position in that. And that's why, without going into too much detail, but you see here the different colors referring to the different situations in the different countries within Europe, whether they allow switching, whether they allow automatic substitution, yes or no. Without having scientific information, solid scientific information on possible interchangeability. In Europe, there is a heterogeneous situation. Also, over the world, you will see that there is some difference in attitude with respect to substitution and interchangeability. And in most cases, there is some reluctant attitude towards the switching issue. And this is also demonstrated nicely by the US-FDA guidelines. On the left-hand side, as I mentioned, EMA does not say anything on interchangeability, whereas the FDA clearly makes a distinction between those two processes. You can demonstrate that your product is biosimilar. That's the first step. You can also set your standards higher in your evaluation of your product, and after having demonstrated biosimilarity, you can also add through clinical data that your product, your biosimilar, is fully interchangeable with the reference product. So you need some extra clinical data demonstrating that switching back and forward between biosimilar and the reference product indeed has no impact on the clinical safety and clinical efficacy profile of your product. So they clearly take a scientific approach and know that biosimilarity is not identical to interchangeability. So these are two different concepts. They have draft guidelines. They are not finalized yet, but you will see here the first thing is, of course, to demonstrate biosimilarity. That's obvious. And then to demonstrate that the product, when administered more than once, that the risk in terms of safety or diminished efficacy between the biosimilar and the reference product is not greater than the risk for the patient when the patient would have stayed on the reference product without any switch. So far as I know, there have no biosimilars in the US been submitted as being possible interchangeable. And obviously, substitution is only, if you look then at the SDA, is only valid, is only allowed when the product has been demonstrated to be interchangeable. So the situation of the substitution is obviously, it's regulated at the state level, but it's only to be considered when it has gotten the label of interchangeable by the FDA. And obviously also in that case, even the physician is allowed to write clearly on the prescription that for some particular of his or her patients, for instance, that the pharmacist is not allowed to substitute, for instance, for whatever reason. Yeah, so if we look at the regulatory approach, I mean, as indicated, especially in Europe, policies vary quite significantly. We should say that in many countries, substitution, automatic substitution, is definitely prohibited. Yeah, for the EMA and for the FDA, I think we have discussed this. Now, the point is, if we want data, clinical data, to demonstrate interchangeability, how will we do this? And I will discuss this only very briefly. First of all, you can have a single switch, single arm study. You have patients that were on the reference product and you switch them to the biosimilar and you evaluate immunogenicity, efficacy, and so on. A better approach is a single switch, but at least a parallel arm, where you have patients that are either switched to the reference product and another set of patients that are kept on the reference product, that then you can really compare at the end whether there is a difference between the patients that were switched and the patients that were kept on the reference product. There are quite a number of other approaches. As you will see here, a crossover study is even a bit better. Also, then you have included the switch from the biosimilar to the reference product. And actually, what is the ideal situation is the multiple switch, where you actually subject your patients to at least 2 switches, from reference to biosimilar or vice versa, and then going back to the original treatment. But it's obviously that there are quite some challenges. You will realize that these type of studies will be very expensive if you have to include this in your biosimilar development. So the financial aspect is at least one reason to be a little bit reluctant to ask for those data. But then the question is, for the sake of patient safety, should we deal with these financial hesitations, yes or no? Another point is, how long should you keep the patient on one treatment before the switch and before the second switch? Is this after 2 months? If this is after 6 months? Is it after 1 year? So this is also a point of discussion, and we are looking forward to see the guidelines, the final guidelines of the FDA, to see what they would actually prefer to see. And without going into too much detail, we have on the one hand scientific challenges related with those switches, again completely different from the chemical molecules, where for instance the half-life is very quick. For monoclonal antibodies, the half-life is 2 to 3 weeks. So when you switch the patient, there will be certain periods where the patient body experiences the originator product together with the biosimilar product, for instance, which may also have some kind of an impact. On the other hand, there are some ethical considerations. You have a patient has undergone already many types of treatment, and then in oncology, for instance, it's the very last possible treatment that is possible for this particular patient population. Will you then take the possible risk to introduce residual uncertainty by switching the patient to the biosimilar, or again vice versa? So this is definitely also a point, a matter of discussion in real life. People will often tell you, or try to tell you, that we have a whole set of data on switching. but I can tell you that most of these so-called lots of data on switching, or people are referring to abstracts on conferences. Patient populations of 10, 15, 20, 50, 60 patients, short term, and so on. So I do not personally consider these data as real, true, scientific, peer-reviewed data on safety of switching. There are two main studies, the NORSwitch study and the DAN-Bio registry-based study. I will discuss very briefly the NORSwitch study, which provide us some data, but again, very limited data with respect to the general statement on interchangeability. And again, very quickly, the NORSwitch study is organized in Norway, as the name says. It's a randomized double-blind study based on switching between the infliximab reference product and the infliximab biosimilar. In total, 480 patients were enrolled, but you should realize that this is a very heterogeneous population, representing, first of all, rheumatoid arthritis patients on the one hand, and inflammatory bowel disease patients on the other hand. So this decreases our statistical power. And again, within the IBD, you have Crohn's disease, you have ulcerative colitis, and also within the inflammatory joint disease population, you have different subsets of disease populations. Again, leading, as you can see here, that for each patient population, you have only 155 with Crohn's, 93 with ulcerative colitis, only 30, for instance, with psoriatic arthritis, and so on. It was a single switch parallel arm. That's one thing. They were followed for 1 year, and the effect of the switch was evaluated with respect to disease worsening, the specific outcome, safety, and short-term immunogenicity, I should say. Now, this is the conclusion that you can find in the scientific paper in the Lancet by Jurgensen. The NORSwitch trial demonstrated that the biosimilar was not inferior. I would say that's fortunate, of course. Unfortunately, the study was not powered enough to show non-inferiority in the individual disease cases. As I mentioned, those 480 patients represented at least 6 to 7 different disease situations. And what is also very important, that notwithstanding the fact that in this particular study, this one single switch from biosimilar, from reference to biosimilar, did not show any extreme situation, devastating situation for the patient. It's clear that these data should not be generalized to other biosimilars. This does not tell us anything about interchangeability or switching for trastuzumab, adalimumab, cetuximab, or whatever other biosimilar. And in addition, what is also very important in the real field, it does not tell us anything for multiple switches, only one switch. And so much more studies will be needed. And this is, first of all, we also will have to take into account traceability issues. Do we know at any time point which product the patient has received? And this will also come back in the pharmacovigilance and naming situation. I won't discuss this. It's also important that we realize that biosimilarity only has to be demonstrated at the time point of registration. We have discussed manufacturing change. Manufacturing change as such should not be a huge concern, but you should realize that at the time of approval, this may be the situation, the reference product here in yellow, the biosimilar in blue, with properties that are overall the majority of the analytical properties are the same, only a few differences, but relative or as a consequence to manufacturing changes, some of these properties may deviate from each other, each within their own specification range. But once you have introduced a manufacturing change, the biosimilar has not to be compared again with the reference product. So for some particular quality attributes, you may have a shift that deviates the two products from each other. And I'm not saying this is obviously exaggerated here, but it just means that we have no idea on the long-term biosimilarity of an approved biosimilar versus the reference. So this definitely is also not the case for an interchangeability property that we have to take into account. Another very important point in real life, much more important than the fact of a one reference versus one biosimilar, is the fact that we should realize that, and I need to remind you to your statistical courses, it's not because product biosimilar A is not significantly different from reference product, and that an independently developed biosimilar B is also not significantly different from the reference product, that we have any information on the possible significance of differences between the two biosimilars. This is purely statistical knowledge. So there is no idea on that. This also means that when two products, A and B, are each independently biosimilar to the reference product, we have no idea whether, from a scientific statistical point of view, A is biosimilar to B. Nobody will ask those data and nobody will perform these experiments. So we can definitely not say that biosimilar A would be interchangeable with biosimilar B. And why is this important? You have seen with the approved biosimilars, quite a number of companies bring good quality biosimilars onto the market. If we have two biosimilars relative to the same product, but independently developed, then the question arises, even if we have data, switching data between the originator and here biosimilar 1, we have no idea whether these data are also extrapolatable for the interchangeability between biosimilar B and the reference product. And we definitely have no information on what happens if we switch between the two different biosimilars. This is just a fact. And this again also in combination with traceability and pharmacovigilance. How will we know when we have, for instance, 4 different biosimilars onto the market, which product each patient has received at each time point, if the competition is so high and if we have many biosimilars onto the market. So that's something we have to take into account for the sake of the patient benefit and also for the sake of the safe implementation of biosimilars. And I think I will stop here. I believe that the summary does not need to be repeated. This is very much conclusion. So I think that for the time being, we have sufficient data that an approved biosimilar is safe to be used in any treatment-naive patient, but we should think very much about how to deal with what I would then call a non-medical switch, purely based on financial reasons between biosimilars and originator, but also vice versa. I thank you.